let us start with the panel. In the panel, I am excited to see Dr. Gus Kusulus. Dr. Kusulus is the president of the National Association of Idea Principal Investigators. He received his degree in physics from the Fairleigh Dickinson University in New Jersey, and his master's degree and PhD degree from Pennsylvania State University in biophysics and molecular cell biology. He received postdoctoral training at the University of Chicago, working in Dr. Bernard Roisman's laboratory at the University of California at San Francisco with Dr. Lenore Pereira, where later he was promoted to research assistant professor. He joined Louisiana State University in Baton Rouge, Louisiana in 1988 and became professor in 1994. He's currently professor of virology and biotechnology at the LSU School of Veterinary Medicine with adjunct appointments at the Department of Biological Sciences, Biotechnology, College of Basic Sciences, the Department of Microbiology and Immunology at the LSU Health Sciences Center in New Orleans, and the LSU Health Sciences Center Stanley S. Scott Cancer Center in New Orleans. He is also an affiliate member of the Tulane National Primate Research Center located in Covington, Louisiana, and he's serving as the head of the Department of Pathobiological Sciences and is the director of the Division of Biotechnology and Molecular Medicine at LSU School of Veterinary Medicine. He's also the principal investigator of the Louisiana Biomedical Research Network funded by NIH and IGMS and other extramural and intramural sources. So, Dr. Kusulus, welcome.
Hey, hello, everybody. Glad to be here. Thank you, Elia. I guess you're going to introduce all other speakers before we can start the panel, right?
Yes, exactly. Thank you very much. Our second panelist is Dr. Sona Vasudevan. Dr. Sona Vasudevan is the director of the Systems Medicine master's degree at Georgetown University. Her interests span in the areas of structural biology, structural bioinformatics, homology modeling, protein ligand and antibody antigen interactions, and RNA biology. Her research interests include comparative genomics, protein classification, protein homology modeling, and molecular dynamic simulations to look at the effect of single nucleotide polymorphisms in breast cancer, as well as evolutionary analysis, systems biology, and translational informatics. She has contributed to the development of several well-known bioinformatics tools for doing sequence and structure analysis. She has developed a drug discovery database, and her research record includes several research papers, invited lectures, and book chapters. She serves on the editorial board of and is a reviewer for several journals. She teaches biochemistry, structural biology, and several courses in informatics, and she has mentored many students and created an MD/MS dual degree program in systems medicine and a freestanding MS degree program in systems medicine. And she has received numerous academic awards and excellence in teaching awards as well. So welcome, Dr. Vasudevan.
Thank you, Elia. It's my pleasure to be here. Thank you for that wonderful introduction. Looking forward to the panel.
Okay, great. Thank you very much. We also have here Dr. Leonard Brodsky, who is the outgoing director of the Tower Bioinformatics Research Center at the University of Haifa in Israel. So Dr. Leonard Brodsky authored a long list of research publications, and his background is in biostatistics, mathematics, and over the past several decades, he has been working in bioinformatics. He has been leading bioinformatics research, computational biology, and systems biology modeling for over 30 years. His work is primarily in bioinformatics algorithm development for many years. And as a result of that work, he has produced a number of new approaches to biomedical data analysis and integration. So currently he is working at the Tower Bioinformatics Research Center and being the scientific advisor to many research projects with collaborators of that center as well. So welcome, Dr. Leonard Brodsky.
Thank you very much.
And also we have today Dr. Raul Andino. Dr. Raul Andino is a professor in the Department of Microbiology and Immunology at the University of California, San Francisco. Dr. Andino is a virologist and professor in the Department of Microbiology and Immunology at the University of California, San Francisco. He is noted for leading a team of researchers that developed the first new oral polio vaccine in 50 years. He has been working in virology for over 30 years and has a broad background in RNA virus biology with focus on molecular biology and biochemistry, viral immunology, vaccine development, and evolutionary biology. RNA viruses are a major threat to human health, and they also provide a fascinating window into processes that can be harnessed to improve human life. So welcome, Dr. Andino.
Thank you. Pleasure to be here.
Okay, great to have you. And finally, we have Dr. Laura Harris. So Dr. Laura Harris is a research mentor and director of training at Michigan State University. Dr. Harris is an educator who enjoys developing curriculum that involves use of computational resources. She also enjoys mentoring others in data science and biomedical research. Her projects use bioinformatics and basic laboratory approaches to examine gene expression changes associated with antibiotic resistance in bacterial species, microbial-induced cancer, and SARS-CoV-2. Dr. Harris is involved with the American Society for Microbiology and has co-authored several educational and scientific papers. Welcome, Dr. Harris.
Thank you. Pleasure to be here this morning.
All right, great. So after all of these introductions, again, thank you everyone for being here. Essentially, what we've done is we've compiled several questions from all of the participants of the symposium that we hope that you can help us address. And so what I wanted to start is, what are some of the changing dynamics around omics data? So essentially, what do you see from your perspective? How is this field of omics data changing in terms of data availability, data quality, data access? And I wanted to first ask Dr. Andino if you could kind of share with us your thoughts on that subject.
Yeah, I mean, thanks, Elia. I think that there's obviously a very important exercise that we need to do in terms of integrating the intuition that many of us have in biology with the intuition that many computational scientists have. And so the first thing that you really need to think about it is how to integrate that kind of perspective and intuition that only comes when a scientist is involved in this type of work for years. So, you know, transmitting that kind of intuition is critical because it really allows you to define the questions and, of course, then define the technical approach to access to the information. And as you know, Elia, because we've been working for many years together, one of the things that's fascinating to me is that every biological process, from an enzyme recognizing the substrate all the way to epidemiological process, they are dictated by the dynamics of the process. I always put this example that if you see a picture of a cat in the air, you really don't know whether the cat is falling or jumping. And you really need a movie, you know, a series of pictures that capture the dynamics, and then you will know the cat is jumping from one place to the other. And so that the same thing happened with biology, and I think the whole thing jumps to, I mean, boils to a simple matter of good data, perhaps with certain temporal resolution that allows to train models that will enable understanding those dynamics. The higher resolution the data is, the better the models will be. But I think the models play a very important role in defining the type of experiments that one can do if one has the opportunity to obtain data that will be more useful to fit these models. then you will be able to have this sort of iterative process in which the models gets better defined based on the data, and the data gets better defined based on the models.
Thank you. Thank you very much for that good illustration with the cat. So I really like that. You really don't know what's going on unless you see the full. So with that being said, my next question would be about what is really the need for multi-omics integration with, genomic, transcriptomic, different types of omics data. What is the need to be able to integrate them, and what are some of the ways that it could be done? And so I wanted to ask that question from Dr. Leonard Brodsky, if you could kind of expand on that.
Yes, thank you. Okay, first I believe that actually we have some difficulties with many technologies in multi-omics data, and the question of integration is how to combine all these technologies, which are giving different results, and after that, how to move from all of that to biology. Second, I completely agree with Raul that time dimension in integration is extremely important, because actually it is saying what kind of biology we virtually can expect from our experiment, like cell differentiation or processes with organism like aging and things like that. And of course, development of virus infection. So two major issues, I believe. How to combine different technologies and how to stream all of that to time dimension.
Okay, great. Thank you very much. So, multi-omics integration, I know that you've been working on that for a long time, but essentially, as also Raul mentioned, this kind of discussion with biologists maybe is really critical. And I also wanted to ask about the ways that omics data is essentially changing research projects. We already kind of heard about basic research, but in terms of translational research, I know that Dr. Vasudevan, you work with a lot of clinicians, you're in the medical school, you kind of prepare students for systems medicine. What is your take on how omics data and some of the things that were mentioned by Dr. Andino and Dr. Brodsky, how is that changing those research projects that you see happening around you?
Yeah, thank you, Elia, for that question. I was hoping you'd ask me that question. And thanks to Dr. Andino and Dr. Brodsky, who already laid an amazing foundation in terms of where we are in terms of starting any research projects. So the way I look at things and the changing phase of informatics is that it's become a very interdisciplinary field. So we cannot I am a physicist by training, converted myself to a biologist and an informatician. But with so much of data coming from all over the place, transcriptomics, proteomics, pretty much, I think there are like 238 or 40 different omic terms and so much data out there. The biggest change in designing a research project is to have an interdisciplinary team. So we need a clinician, we need a biologist. In fact, we even need an economist now to see the different ways to do things. So the way, even when I teach medical students, so we talk about one disease, one gene. So that's no longer in play right now because it's like a whole systems approach. Different genes expressing different organ systems, different cell types, different things. So it's a whole systems approach. So what is really changing with all of this is, of course, multi-omics integration is crucial. And the most important is also interdisciplinary collaborations with different expertise that is involved. And that's what I teach the students. And we have a team now with different expertise. And I'll be talking more about the program at Georgetown tomorrow, but that's pretty much how we have designed so that there is a lot of collaboration across different fields. And that makes multi-omic integration more efficient as well.
Okay, great. Thank you very much for that insight, Dr. Vasudevan. And, another thing that I know that is a challenge in some ways with this kind of dynamics of omics data is essentially how is it practically used? So I know that there are hubs where, omics data is available and everybody seems to know how to use it. And then there are places where there's a need for training, there's a need for access, and there's a need for kind of elevating the level of biomedical research. And I know that Dr. Kusulus, you're involved with Louisiana Biomedical Research Network, with the National Association of Idea Principal Investigators. And so I wanted to ask you a question about how important is bioinformatics in many of the research projects that you are involved in, as I'm sure a mentor and a reviewer for many of these networks. And how do you see the trajectory of kind of data science and bioinformatics as a critical need in many of the projects that you oversee?
Yeah, thanks, Elia. Elia mentioned NEPI, the National Association of Idea Principal Investigators. Just to give you a very brief description, this is a nonprofit 503c corporation that represents 23 states and Puerto Rico. The program is funded through NIH with over $400 million per year in multiple centers for biomedical research excellence that relates to promoting people into independent funding through the R1 mechanism. Every state has the IMBRI programs, the National Idea Biomedical Research Excellence programs to promote biomedical research. And those IMBRI programs for every state have a priority for data science and bioinformatics. And of course, we have the entrepreneurship hubs and the clinical translational centers these are the 4 programs that are part of NEPI. There are over 170 programs and principal investigators that are members of NEPI representing those 23 states in Puerto Rico. And as already alluded to, NIH has recognized the importance of data science and bioinformatics. In fact, there is a complete division of data science, and the teaching and training and providing support for data science and bioinformatics is now mandatory for all the IMBRI programs for those 24 states. And of course, it's extremely used into the clinical translational centers that are funded by NIHGMS, as well as by other categorical institutes, and the COBRI programs, the Center for Biomedical Research Excellence. The NIH recognition of this importance is really multi-level because now they have arranged with Microsoft and Amazon and others to really utilize their resources to be able to really do high-level computation in the cloud as opposed to locally. And of course, there are many, many databases that are now supported by NIH for clinical translational research. The goal of NEPI, as well as all the IMBRI other programs, are really to reach out and prepare faculty, staff, and students in dealing with bioinformatics. And of course, as I was alluded to a couple of our speakers, I'm also a physicist that turned into molecular biologist. And my view of the world is that really everything that we look at in biology, which often is descriptive, ultimately has a physical explanation to it and a defined parameter to it. Every process that we do, whether it's receptor binding or anything internal to the cell, it's actually described, can be described by some physical formula. And ultimately, this physical world really translates into tremendous amount of information that cannot really be handled through a descriptive way of understanding what's happening within that cell, tissue, or organism, or even in population as we do that. And that's where we need the computational tools. And the approach that we use is really not only to have interdisciplinary teams, which obviously at the level of higher education is a solution. But even more importantly, to really teach young students, undergraduates, graduates, the basic tools of data science and bioinformatics through intensive exercise programs. Because I think ultimately we need interdisciplinary education at all levels, because the new generations need to be trained to understand and utilize these tools. And the quicker and earlier we reach out to them, the more important would be for them as they pursue biomedical research. And of course, this is extremely needed. I think 90% or more of every research grant I reviewed, not only within the LBRM program, but the national programs, whether it's cancer research or infectious diseases, now use advanced transcriptomics, RNA-seq, proteomics, integration of transcriptomics, proteomics, and genomics. So it's a must tool now, it's a necessary tool to be able to compete at a higher level.
Okay, thank you very much, Dr. Kousoulas. And my next question is to Dr. Harris, who I know is somewhat of a recent convert to the domain of bioinformatics and also comes from a biology background and now deals with a lot of student projects as a mentor and runs a whole program. And so I wanted to ask Dr. Harris, what are some of the ways that researchers and students can prepare to deal with this increasing volume and complexity of omics data in research? How did you yourself come to the need for bioinformatics, and how are you using it now?
Great question. I came to need bioinformatics mainly because I ran out of options in a wet bench setting. So wet bench tends to be very expensive. It can be very time consuming, and it generates a limited amount of data for usually the money that you're putting into an experiment. By using things like computational meta-analysis approach, you can then take different experiments across different labs and then synthesize new information that may be missed if you're just looking at a couple of samples or a couple of mice in your own lab. But that can be very overwhelming. And it was overwhelming for me when you see how big some of these data sets can be when you start aggregating them across different platforms or knowledge bases. So my advice to new students is really be organized. And I have come to find myself falling back on the traditional lab notebook that I was trained with in the wet bench. So, we're trained, we have a lab notebook, we go into the lab, we document everything that is done that day, procedure, results, corrections, thoughts on tweaking the procedure, that sort of thing. The same thing applies to bioinformatics. So you have to treat it just like you would a wet bench research project. You need to have a notebook that says, I tried this approach, it worked, it didn't work. Why do I think it didn't work? What am I going to change next time? Because when you start layering all these different omics, or for me, multi-omics on multi-projects, it can be very confusing and overwhelming and even intimidating to see just the amount of data, and having that notebook to kind of help you break down, okay, here's where that project is, here's what we did, here's why something needs to be tweaked the way it is helpful for my sanity, helpful for my students' sanity. And it's also nice to then have a global perspective across projects. So if one project encounters a problem and I forget about it, but then 3 weeks later, another project comes into a similar problem, I can then go back through the notebook and say, oh, wait a second. Yeah, I remember how we fixed that here now. Why don't we try it on this new project? And hopefully you get lucky.
Great, thank you. Yeah, so I think that is very useful to kind of keep track of everything. And, that brings me to my next question. And this is a question that I wanted to start with Dr. Andino. you've been doing virology for 30 years, as I've read in your background. I wasn't around maybe back when you started, but how has the types of data and the ways that this data is being used, how has it changed in recent years for you, for the types of research that you and your lab do? You're on mute.
Yes, it's obvious that the process, the data that we're using, as a little bit of what Dr. Harris was saying, it used to be that you will run an experiment and you will have a specific answer to a particular process that is well defined within your experimental paradigm. And this has changed enormously because now you are able to use a lot more data points and a lot more information from the same well-defined experimental paradigm. And the question is how you interpret that. And, we've been discussing about this, this ability to, use this data to, try to extract more information to this particular experiment. But I think what really is transformative is what Dr. Vasudevan was saying at the beginning. What we're doing now is, and we see this very clearly on this process, what we are in the middle of this pandemic, is that many of the things that we need to do now require the integration of different experts. And I just wanted to say that the omics is actually an interesting process because science is a human activity. And one of the things that we encountered recently is that the most important thing you can do is to form a team that can create its own culture so it can sort of interact in an effective way. If you need the expertise of a clinician, virologist, a computer scientist, an epidemiologist, an economist, then you really need to have a common language. And so in my mind, one of the things that is changing now is that we're starting to, generate some sort of culture of discussion. And this symposium, which I'm very excited to participate, precisely because you are bringing young people that recognize the importance of this kind of omics and integration. But in my mind is what's going to be the future scientists. The scientists of the future is where you really create a person that, or a scientist that is not afraid to reach out, collaborate, and use all the information and intuition that our experts have developed over years. I'm not sure if I addressed the question, but to me, it's one of those really important things that happened to us in recent years.
So thank you. Yeah, maybe not a direct answer to the question, but a very meaningful kind of addition, I think, to the need for interaction, for the need of common kind of understanding and that ability to listen to the different sides of the story to really come up with results. So I think that was very useful. So thank you for that. My next question is, about the necessary skills that you look for when you are looking to add a bioinformatician to join a project. And so I know that, in many areas, bioinformatics is such a kind of a diverse term. It could mean something very simple. It could mean organizing a database. It could mean processing some data, or maybe developing a completely new approach in an algorithm. So it means very different things in different kind of fields. So I do want to have all of you, kind of add to the answers. But my first question is to Dr. Brodsky. You lead a bioinformatics research center. you develop novel algorithms and you kind of identify solutions to problems that you see biologists struggle with. What are some of the necessary skills that are most important, in your opinion, for new bioinformaticians or new people joining your lab or your center?
Yes. Okay. Actually, one cannot expect all kind of expertise from a person. So it's better to have several people with different majors, so to say. So I'm expecting good programming skills and maybe high level of programming and algorithm development from one type of people. Another type of people are expertise which have expertise in mathematics and statistics. This is not always the same person is a good programmer and a good mathematician. And actually some basic knowledge of biology, because in biology, we rely on our collaborators, not on ourselves. And so altogether, and a good contact with collaborator, actually, this is what is needed for the team to move forward, as I believe.
So thank you, yes, for that answer. So I think kind of similar to what Dr. Andino also mentioned is that need to combine people from different backgrounds and different skill sets and enable them to work together. And so in the same direction, I also wanted to ask Dr. Kusulus, you try to work with these different programs to not only train and build up their capacity, but eventually also to kind of solve bioinformatics challenges in a research project. So what are some of the things that you look for when you're hiring, a bioinformatician, or when you're looking to include a bioinformatician in the research work that you do?
Yeah, that's a very relevant question, Eli. Thank you. Obviously, if you're within certain institutions, you may not have the capacity to build a team of 3 or 4 or more bioinformaticians that represent different aspects that are needed for collaboration purposes. So the way we recommend and we are using this is to really get only few people involved as liaisons to resources and talent on the outside. So we look to hire a bioinformatician that has obviously computing strength, some statistics, understands pipelines. We're using a lot through our Illumina-based core bioinformatics for infectious diseases. RNA-seq is the principal tool. We're now going into metabolomics and proteomics data. And the idea is to really have 1 or 2 people. We do have a PhD, for instance, in biostatistics. We'll be hiring a bioinformatician. We had one. We lost him to the University of Wisconsin. But the idea is to really liaison with resources and collaborators on the outside. So we have an MOU with the Tolbert Bioinformatics Research Center in Israel with Dr. Brodsky for potential collaborative projects. And of course, we work with Pine Biotech to utilize their training tools and pipelines. So our accommodation is really for high-end core facilities like ours that have supported a number of NIH projects to have 2 or 3 people internally that are broadly educated and then link up to external resources, including really commercial resources that are available for a fee-for-service to provide support for that. But even beyond that, I think it's extremely important what I mentioned earlier. We like to push the ability to handle data at the lowest possible level. So intensive training of graduate students, undergraduate students that are in computer science, that want to learn biology, biologists that want to learn data science, bioinformatics. Part of the idea is not to get only the PI to collaborate, but have the actual laboratory with the people on the bench be able to use bioinformatics tools, liaison with a more expert person, and then utilize the external resources, whether commercial, collaborative, or NIH resources that could be doing this, because it would be very difficult for us to hire a big team of people to handle some of our projects.
All right, yes, great. Thank you very much, Dr. Kusulus. And Sona, I know that you as a researcher and especially as a director of a program, I know that students come to your program from all over the world with all kinds of different intentions. You know, your goal in just a short time to train them for what will be expected. So I know that you're probably feeling out there in the industry and the research, you know, what are the skills that people are looking for in a bioinformatician or somebody that does systems medicine? What do you think are some of the necessary skills that you're looking for them to acquire and be graduated with to make them ready for that next position?
Thank you, Elia. I've actually must confess that I've lost sleep over this just because it's not practical and it's not easy to create a bioinformatician, ideal bioinformatician who has lab skill sets, statistical skill sets, and understanding data. And at the end of the day, we are all trying to solve problems which are biological in nature. And so it's a very challenging thing. And I do get students with different backgrounds. I have students who have psychology backgrounds. I, in fact, had a student who had a history background. So it's a very challenging thing. But in the least, what I try to do is give them some basic foundation of the biology and the questions that we're trying to address. And then give them a broad skill sets of a little bit of statistics and a little bit of programming skill sets. But unfortunately, not everyone has the acumen or you know, the mindset to grasp everything. But the program, what I try to do is just to expose them to different aspects of informatics out there. So they don't come out as hardcore software engineers, but at the same time, they have basic foundation that they can build on to develop their software skill sets if that's what they want to pursue. Or if their students want to go and work in the bench to, you know, bench side, then they do have exposure to understand how to extract RNA and how do you prepare the samples to send for sequencing and do RNA-seq analysis. And they do have an overall, all of them actually walk out with the you know, aspect that we are all drowned in data. So how do you look at data? So statistics is a big part of it. And so they do learn statistics. So students, after they graduate with skill sets in different areas, then they decide where they want to develop more. And so, you know, some of them, you know, take courses and electives to make some of their foundations more strong based on their interest. But one thing that is important in the program is that they all do research. So capstone project is you know, is mandatory for them. So that's where they kind of tie in together and become experts in one small thing. Some of them are interested in programming, so we find mentors who you know, engage them in learning programming, and some of them want to do bench work. So it's all over the place, but at the end of the day, they develop a little bit of wine tasting aspects of different things, and then they go ahead and develop whatever skill set they need. Of course, long story short, I think I would, if I were to hire someone, of course, I don't have, you know, humongous amount of money to hire a statistician, a biologist, and all of those things. So with limited funding, we just have to pick a student who has little bit of understanding of programming and biology for sure, and then who embraces the use of software tools, resources, and you know, understands how to put together data in the context of biology.
All right, great. Thank you for that perspective too. And Dr. Harris, you know, the same question I wanted to kind of rephrase for what I know about what you do. I know that many times you deal with students that have maybe very limited background in bioinformatics, and nevertheless, you're able to integrate them in some of the excellent projects that you do, providing them with a meaningful experience. And I know that you also put out a number of resources out there that are facilitating that process of getting people interested. And so I wonder, what is your take on how to essentially get started for somebody that wants to get involved in a meaningful research involving bioinformatics? And how does that experience prepare them for what real bioinformatics needs out there exist?
Yeah, I was wondering if I was going to get a question variation simply because my answer to the first question would be radically different than the other panelists. I'm one because, as you said, I don't get a lot of students that have expertise. I've never hired an intern. I've never hired a graduate student for research. That is, I hire them as teaching assistants and other things like that. But for me personally, it's more about the personal characteristics of the student. Are they curious? Are they curious enough to go find an answer rather than wait for me to give it to them? Even if they don't know where to look, at least trying gets you somewhere. Or if they do run into a problem, are they going to shut down and not ask questions? Or are they going to come forward immediately and start asking questions of their fellow researchers, of me, of whatever people they run into at conferences or what have you? So a lot of those characteristics are more along the lines of what I look for rather than I'm looking for someone in stats, or I'm looking for someone in comp, or I'm looking for someone in bio. Because for the students I get, which can be anywhere from basic high school all the way through graduate level, they don't necessarily know what they want. They don't know what they're interested in. They don't know where their anxieties are. And so coming into my research lab or my trainings, frequently that's an experience they're learning about themselves. And so I get to discover that alongside them. Sometimes they get into a bioinformatics project and they find, the computer really isn't as exciting as I thought it would be, or the lab is not as exciting as I thought it would be. And so things like, flexibility, perseverance, attention to detail, cannot stress that enough, are all very important things that I look for rather than looking for a skill set. I can train a skill set. I can train a biologist. I can train a computational scientist, but I can't train curiosity. I can't train attention to detail. And that's really where I need the students to step up and bring those characteristics to the plate.
Okay, perfect. So thank you again, Dr. Harris, for your perspective and for everybody that shared all of this wonderful information. I do want to mention that we have a link, and maybe Ojasvi or Sonalika, you can paste that link, where we have profiles of all of the panelists and speakers, where you can find links to their websites, to their programs. You can learn about their background, and you can obviously, connect with them if you would like to, or they would like to accept that request. But I hope that this not only provides us with some perspectives, but also leads to other outcomes. Now, I want to briefly introduce another person that wants to, that can add an important perspective to this. So Gio, or Gipoliano Chavez, he is a mentor at the summer internship program run by UCSD, but also is starting to mentor some of the students that come to our research fellowship program. And I wanted, Gio, to ask you if you could add a little bit of your perspective on how, at the very early stages, how do you help students, essentially get interested and do something meaningful with data. And I know that you lead some projects on your own, but also you kind of became converted to the field of bioinformatics not so long ago. So maybe you could give us a little perspective from your side. What do you think about this?
Yeah, I was particularly interested when Dr. Laura Harris mentioned that about the curiosity of students. Is the person curious? Are they curious about diseases, or what the phenomena that have led to a disease in their family, for instance? So a lot of countries have problems in allowing access to science resources. For instance, I come from Brazil. I come from public schools in Brazil, and there are lots of limitations in the country there. So I came as part of an effort that the government of Brazil is doing right now, or has been doing in the past decade or so, to launch science, or to try to cultivate a culture of science in the country. So we have collaborations. We have I have been, as Elia mentioned, I have been serving as a mentor for a program called Science Internship Program at the University of California, Santa Cruz. And the mission of that program is to really bring mainly high school students, but as I turned to realize, it's expanding from only the scope of high school students to become to be in contact with scientists, with mentors. So what SIP, the Science Internship Program, does is it brings high school students to shadow PhD students. I think there might be master's students that also serve as mentors of SIP, and that really makes the student think, I really want to pursue a career in science, or I definitely do not want to pursue a career in science, because that's also important to have that decision, and having the resources to decide on whether or not pursuing a career in science. So I think it's really important to, as early as possible, start to educate kids and young scientists. Young scientists may be not a very good way to put it, because we just want to make sure and guarantee that kids have a chance to understand what science is, and have a feeling of what working in a science lab, in a research institution, what it looks like. And I think it's really important that in countries that do not have an economic situation like the United States, or the so-called first world economies, I think it's really important to allow these other countries, these other regions, these other communities to build this type of experience for their youth, for their young generations.
Thank you, Gio. In summary, I just wanted to ask all of you, all of the panelists, to maybe, if you want to add anything to what we discussed in this panel. We do have a few questions from the audience that I will read out in just a couple of minutes. But before we do that, I wanted to give you an opportunity to just maybe say a couple more things from your perspective. Again, the topic of the panel is the changing kind of need of bioinformatics and the changing kind of technologies in this field. We will continue to hear about it in more in-depth talks in just a few minutes. But please, does anybody anybody from the panel want to add something to this topic from what we discussed so far? Dr. Andino, would you like to add anything to this?
There's been a tremendous, exciting discussion. I think we all in the panel recognize the importance of bringing, not like Gio was saying, young people, everybody, I would say newcomers to these activities. I think that will be incredibly important if, really this symposium brings people together to realize the power and also the challenges in this exercise. I mean, I guess the only thing that I wanted to say is, just emphasize that it really is a human activity. Is bringing together biostatisticians with empirical scientists, if you will, and together to these discussions. The curiosity point is very well taken. I think we all need to emphasize that, that is something that is essential to the process. And I like this idea that you create this circle, iterative circle between, data analyst and experimentalist, because, if you think about it, you can have a big data set and discover something that is inspiring in terms of creating a new model, a new perspective of how you see something. But at some point, you need to validate that observation. And how do you confirm that observation is real? Well, you need an experiment. So I think, again, this is a team effort. And it's, my personal experience working with Leonid Brodsky and Elia Brodsky, all this last 10 years or so that we've been working together, has been that I learned a lot myself, educate myself by these interactions. And I think that's very exciting from my perspective, the possibility of continue learning and not being so young, but a newcomer.
All right, thank you, Dr. Andino. That's a great perspective there. And Dr. Leonard Brodsky, would you like to add anything?
Okay, bioinformatics is becoming very complex. Actually, the entire science, from mathematics to quantum computers, is involved in bioinformatics. And also biology itself is developing very fast. So a lot of challenges, and actually all challenges should be covered by education. And actually the education has to be a little bit in front of challenges, say. And I believe that education is very substantial part of bioinformatics in general, and not only bioinformatics, but this joint venture of bioinformatics and biology, because without biology, bioinformatics is nothing.
Great, thank you. Thank you for that perspective. And Dr. Kusulus, would you like to add any final words to this discussion?
Yeah, obviously, I think we're all in agreement of the importance of these fields and the challenges that we face. But, in a simple way, advanced data science bioinformatics really can find a needle in a haystack. But the way that Raul indicated, ultimately, you have to really look whether that needle is actually doing something. So it's a unification of really the data science bioinformatics and ultimately experimentation that will give you, the answers to what you may need. So we need both. And I can tell you, because I've served as associate vice president for Louisiana State University for almost 7 years, and I ended up reviewing a lot of engineering and physics projects, astrophysics, fluid dynamics, mechanical engineering. And I tell you, there's a lot of solutions developed by other fields that have not yet really fully applied in biology. So I think this merging of disciplines is required, and we need to educate the new generation students. Most medical schools and most veterinary schools have practically very little of education towards this, MDs and DVMs for genetics, genomics, and the use of these tools. So we need a transformative steps in higher education to be able to really train the new scientists that will be able to use these tools. Because these tools cannot be only at the hands of experts. Extreme need that Dr. Brodsky mentioned to really educate people.
All right, great. Thank you very much. Dr. Vasudevan, any final words?
Yes, I agree with all the panelists, and I think education is the most important thing. It's not just educating students, but also educating faculty who are in different fields and who are still scared to come and use data and understand. So I feel like the education should spread to a lot of faculty as well in an institution, in addition to students, because I have several faculty on my own, my own colleagues who are either scared or who are skeptical just because there's no knowledge of how to use data. They all have lots of data, and now I think everyone will agree that this is a new language, and I think this language has to be learned by everyone. And I think as Dr. Kusulus just said, we need transformative educational programs, and I think Elia, Pine Biotech, is actually leading in that. I really like how we are putting together, and I also agree with Dr. Chavez that the education should start at the high school level. Sometimes what I teach my graduate students, I do teach high school students, I mentor high school students, and it's amazing to find that sometimes they're smarter, come up with better ideas than graduate students. So I think that there is lots of thirst right there, and all the school systems have to integrate informatics starting from high school, undergraduate, medical school, of course. I'm trying my best at Georgetown as well. So I think this is a very important language that everyone should learn at whatever career paths that they actually take. Things have changed.
Excellent, thank you. Dr. Harris, what would you like to add?
Well, I echo what the other panelists have said about how the students, the graduate students, the postdocs, and even the faculty need to be trained in interdisciplinary research. And that is a challenge, particularly for us old farts who are stuck in our ways. With that said, it is a challenge for students coming up because it seems like they need or feel the need to pick a topic. And I really just want to encourage people to follow their interests, follow their curiosity, follow what drives them, because you never know where that's going to lead you. And one day that could be something that's specifically biology, another day that could be something that's more computational, and then a year later you begin to realize how they integrate together. So just don't let yourself limit your potential. And just keep your head up and moving forward in as fast of a positive direction as you can.
Excellent. Thank you, Dr. Harris. Indeed, very inspirational to continue. And finally, Dr. Chavez, would you like to just add a couple more words to this, just from your perspective? One sec, let me unmute you. There we go.
Yeah, I agree that science is an activity, is a human activity that we, as humans, we should all be engaged in. I just think that it's a personal, not a personal, it's an effort, I guess, that all of the countries and all of the societies need to do in engaging their populations in this activity that can bring benefits for the societies in general.
All right, great. Thank you, Gio, and thank you all the panelists. I want to mention again to everyone that we have profiles of all of the speakers right here. You can find all of the speakers and panelists with their background. You will find, if you click on these images, you will actually get links